Thank you very much, Nadine. And as you say, let's move now into the discussion, the panel discussion about clinical development plans and the regulatory milestones. And to help me do that with the panel, Fabrice is going to continue with us, but I'm joined by two colleagues, both from the New Deal, Daniel Perez and Patrice Marche, from INSERM, to join me in the discussions. And I've got a list of questions that I want to put to the panel. And what I plan to do, I've got a couple of slides where I'll when we start, I'll just say a few introductory words, and then I've got questions to put to the panel, which we can all discuss, and then I'll open it up for questions as well from everybody else who's on this in this workshop. And of course, we've got the chat room as well that you can put questions. So Fabrice has introduced himself, but Danielle, do you just want to introduce yourself and just say a few words about your background so everybody knows where you're coming from?
Yeah, so briefly, I'm Daniel. I'm the Chief Innovation Officer at Nanoimmunotech, an IT partner in New Deal. In the project New Deal, we are in charge of the safety assessment and also characterization of the nanomaterials. And briefly, I started with toxicology in 2004. And then in 2012, I focused only in nanotechnology. So my expertise is how to applicate or find solutions in the characterization of nanoparticles and safety mainly.
Okay, thanks, Danielle. Patrice, do you want to introduce yourself?
Yeah, hello everybody. So I'm Patrice Marche, working in Grenoble for the Institute of Health in France. So I am an immunologist. I'm an old immunologist. I got my PhD a couple of years ago in '81. So most of my career is in immunology, but starting the 21st century, I became interested into nanotechnology, which was developed at the Sevres site where I was located at that period of time. And so I began to be interesting and first put the first foot in nanotechnology by participating to the Nano2Live network of excellence between 2004 2008. And then I got contaminated by this very challenging field, and I participated to several projects thanks to the participation of the different colleagues. And now I am involved in this exciting project with Fabrice and my colleague in New Deal.
Thank you very much, Patrice. I'm just going to share my screen now and just show a couple of slides by way of introduction. Boom, boom, there it is. Everybody see that? Okay. Now, I showed this slide when during the introduction. I'm not going to go through it again, but just to emphasize really that we're going to be covering most of these aspects and talking about toxicology and some of the issues there and the clinical issues and the regulatory issues. We're not going to spend too much time on manufacturing because there's a whole session after the break relating to manufacturing. But if there are questions, we can always pick those up. Now, by way of introduction, I mean, we've seen now, and Ruth presented very well, the fact that we do have already approved products that are nanomedicine based. But if you look at the list of products, and there's about 50 or 60 of them, most of those molecules, products that have been approved, are actually what I call chemically based. They're using new chemical entities, use those, and they're particularly in oncology. But the world's changing, and as we've seen, I think, from the projects that have been presented so far, we're moving to a much more biological base for treating medicine, for treating patients. And around 80% actually of all the medicines in development, if you talk to the FDA and the MEA, about 80% of all the medicines currently in development are biologically based, be it peptides, immunotherapies, DNA, gene therapies, RNA-based products. So our approach to treating patients is changing and has done over the last 15 years and will only continue to do so. But of course, one of the problems that we have in delivering these biologicals is getting them delivered to the site of action. And it usually is a problem for many biologicals. It's a bit like where you're buying a house, really. It's all about location, location, location. And how do we get these treatments and therapies to the sites where they're needed? And this, of course, is where then nanomedicines in particular come in. So if I just stop that and stop sharing my screen. So with the team, perhaps I could, and the panel, perhaps I could put my first question to you all, and then we'll join in on this. What do you see is we've all been involved in developing what I call non-nanomedicines in the past and bringing those forward. What do you see are the main differences between nanomedicines and what we're dealing with now and non-nanomedicines? Anybody want to start on that? Daniel, do you want to have a go?
Yeah, first, I start. Yeah. I start.
Yeah, you always start.
Yeah, I always, yeah. So as I said at the beginning, I start with non-medicine therapeutics. And when I firstly found the nanotechnology in my field, I found several problems. The first problem is that you cannot use the typical assay to characterize your drug, because there is a new actor in the complex. Nano component is a big actor, and it's not only a marginal actor, but it's also needs to be described and also needs to be characterized, and it's also interfering with some common assays. So the first challenge is how we performed experiments, take into account the nano component, and also how we address the questions that we want to answer, having in account the influence, the impact, or the inhibition or activation that can have this nano component in the characterization of the final product. So the first challenge is to sometimes create or performing a special setup for the nano component, and also the combination of the drug, the active molecule, and the component. So first, I do in my talks, I always say that nanotechnology is new cars for the drugs. But recently, I heard a talk that the similarity was with a fire truck. So in a trunk, you can have not only firemen, but all other things, but also the trunk has activity and can have some effects. So it's a new actor that you need to characterize and you need to understand very well. And there is also a need to create new tools to characterize and new protocols, new assays, I would say, to characterize the nano component and the combination of the active reagent, the active agent, and the nano carrier.
Okay, thanks, Patrice. Fabrice, do you want to add anything to that?
No, I think that combining drugs and nanoparticles exists for a while now. But with biological, I think we have several specificity in terms, because when we work with siRNA, for example, we are more vulnerable to the biological environment, to RNAs and so on. So the cargo is not only to deliver at the site of action, but it's also a matter of protection of the biological ingredient. And moreover, you deliver in a site of action where we cannot go without the nanoparticles. It's the case in New Deal. We target the immune cell, so we know that without the nanoparticle, we cannot deliver the siRNA in this human cell. So the siRNA will stick to the mucus or will be degraded before to reach its site of action. So I think that the nanomedicine field is quite old. We enter in the age of maturity, as said by Roots. We have plenty of nanoparticles, and maybe some of them are really relevant for several applications. inhalation, pulmonary disease, and so on. But also there is the biotherapy is booming, especially with the example of patisiran for siRNA, but not only protein therapeutics, peptide. You mentioned also gene editing. Maybe we can also talk about CRISPR-Cas9. You know, it's quite in the field today. So I think that in crossing this way of nanomedicine and biotherapy, we have new opportunities and maybe new attention to pay on that.
Okay, thank you. Patrice, what's your view on how different it is?
Well, a lot of things were already said. What I want to highlight is what the difference between nanomedicine and non-nanomedicine was your original question. To my point of view, the first difference is the fact that in nanomedicine we will compile together different structure, different molecular structure with different capacities. So we elaborate materials, smart materials with different properties for different action, which is relatively rare in terms of designing a simple drug. So of course we mentioned the delivery issue. We can also add imaging if you want to control the targeting and so on, and also you want to have efficient drugs and so on. So really what's characterized the nanomedicine and it's the combination of different action with different materials, and that's complicated a lot the issue of mastering, of course, the fabrication process, but also about the regulatory purpose and the safety. The second point I wanted to add to this is I forget it. I will come later. I'm sorry. I was so enthusiastic to what I was saying. I forget my second point.
Don't worry. We'll come back to that. I mean, one of the things that struck me since I've started working, with nanomedicines, because, I've been in the industry 35 years. The first half I was just developing chemical agents. And now, of course, being, with gene therapies and everything else that comes along. But the thing that strikes me is that when we're developing nanomedicines, we need to start very early on using that nanoparticle, even for the basic research, to deliver it. We, unlike the chemicals, very often we'll get well into humans, say with solutions or capsules, before we end up with the final formulation. But we can't do that with nanomedicines. And, we start looking at them a lot earlier on, basically the formulation and how we're going to deliver it. So that's what I see is one of the biggest differences. And clearly it brings its own challenges to that.
At the same time, perhaps if I may add a comment there.
Yeah.
It also offers an opportunity in a sense that once you have a working capsule or a working nanoparticle ready, you might be able to follow up on that very fast. I mean, once you've mastered to deliver one siRNA, as Alnylam is now showing, the second one becomes a lot easier. So although the first hurdle is indeed tremendous, after that, there's an increase in speed, I'd say.
Yeah, and I entirely agree with you. And also, it depends what we're putting in the nanoparticle. Because, I think, certainly putting in, say, some of the oncology agents that were given intravenously and orally and then into nanoparticles probably went a bit faster. Mm-hmm. And again, from my experience in the gene therapy, when we started 20 years ago, there were no regulations and everybody was very scared about it. And, we didn't know what to do. Mm-hmm. But over time now, we've got products that are approved and it is a lot quicker. So I entirely agree with you there. So if I just go back to the panel, what do you think from your perspectives, the advantages are of using nanoparticles versus non-nanoparticle medicines and what they bring for the patient?
I think the first advantage is that with the same active molecule, by different ways of combinations, you can have new effects new targets, and also new behavior, new toxical profile. There is described how drugs combined with nanoparticles have a different behavior in a tox view, because the toxic part is not more available to contact with the cells and then damage the cells, because they are commonly bind to a structure. So it's opening the way the equation is a new factor in the equation. So the combinatory is exponential.
Yeah, and do you think, Danielle, from your experience, do you think we'll come and talk about what we need to do in terms of toxicology assessments in a while, but do you think that what you're seeing from a toxicology aspect when you're putting those through, that actually you see less toxic effects using nanoparticles than you would do with a normal agent?
In some cases, yes, because it's because the molecule is not available. Yeah. You know, if it's encapsulated, you don't have it available, so the toxic effect is not acting. But once the drug is released, you can have the same effect as the free drug. So, but in sometimes you have an increase of the toxicity,
right,
due to the synergy, also of the distribution.
I see you being joined by a little one, but
yeah, it's a
Don't worry. Patrice, any comments on it?
Yeah, well, I think the great advantage of nanomedicine is what we developed before, is the combination of different functions together. And really, that's a dream. So, but what is a dream is to be able to do everything with a single entity, okay? But that's also, that's the dream for an engineer, but that's a nightmare for the people who are concerned by toxicology. Because more they put action, more they put stuff on their done in nanoparticles, then we have many questions rising. Okay, I see Daniel laughing, because we share some of that concern in the consortium with very dynamic colleagues who produce plenty of nanoparticles, but it's a nightmare for the people who are wanting to master this issue of toxicity.
Yes, and characterize them. Fabrice, what do you see are the advantages?
So as Daniel said, I think that the main advantage is targeting, because you can really have the maximal dosage of your active ingredient at the site of action you want to target. So, and you limit by this way the content in other place in your body. So that really the targeting first, but also through the encapsulation or the loading of your drugs, you can also consider new therapeutic strategy. This has been underlined by Roots during her introduction, but you can combine several drugs and have, let's say, that synergistic effect at the site of action. That's also another opportunity given by the nanomedicine.
Okay, right. Now let me just change the subject. There are regulatory guidelines now. There's both the FDA and the MEA and other national authorities have issued. But one of the problems we have is once they're written and the speed that things are developing, they often become quite out of date quite quickly. That's one of the issues that I see, particularly with a lot of the new advanced therapies that have come out. Do you think that with what we've got in terms of the regulatory guidelines, do you think they're up to date? Do you think they're helpful? Do you think it's time to have a look at them and revise them? They don't really deal with a lot of the biological approaches that we're taking, certainly from my perspective. Any views on that? And again, Daniel, you're probably closest to this, aren't you?
Yeah.
You know where to start. Go ahead, Daniel.
What I would say is since 2018, there is a specific guidance from the FDA for liposomes. But the thing is that it's amazing the differences between nanoparticles. So it's very difficult to put everything in the same box. Even with liposomes, with the same manufacturer, you can have amazing different type of liposome because the impurities are different, the charge are different, the size is different. Any slight difference makes a new component. So what I wanted to say is that the questions from the agency are the same. The only differences is that we don't have only one actor. The first actor is the active molecule. So there is the questions, the purity of this active action, the benefit of this active action, the efficacy. And now we have with nanomedicine and second actor. And for this actor, we should answer the same questions and describe the nanoparticle, describe to characterize a physicochemical characterization of this nanoparticle, know it very well, and also explain the advantage to the agency of combining this component with the active drug. And it's more complex than a formulating agent. So we can think that nanotechnology is a way of formulating is more than that. It's more complex. And sometimes it's very difficult to answer to some questions. I had the experience with a customer that the customer told me, okay, the agency told me, where are the nanoparticles? And I don't know how to answer this question because we don't know. So how to perform the experiments, how to create a tool to try to see the biodistribution of this component, to answer the questions of the agency, because they wanted not only to know where the active molecule is going, but where the nano component is going also.
Okay, thanks. Patrice, any views on this?
Well, in that matter, I would say that's the role of the academic science to help to design the future rules and recommendation. It's obvious that it's not easy to draw at once wise recommendation for the use of all kind of nanoparticles, because the number of combination you can create is definitively very, very large. So I will say defining the regulation, it's a continuous work where we have to have crossfit between the advance in the mechanistic involved in the reaction to nanomaterials and the regulatory agency and so on. So it's definitively so. To your question, is the regulation satisfactory? I will say no, but the way to improve is tight collaboration between basic research and regulation.
Thank you. Fabrice, any comments?
No, I have no further comment on that matter, but maybe I would to comment one of the questions put by the attendees. Fanny, so Fanny is connected. It's nice to see that you are there, Fanny. And you have a comment about the FDA evolution. So maybe Fanny, you can directly comment your point.
Yeah. Fanny?
She only can if somebody helps her to.
No, I'm the host. So
okay.
Hi. Good afternoon. Can you hear me now?
Yeah, Fanny.
Yes. Hi.
Nice to hear you.
Yes, it's just want to mention that I am aware that FDA is now starting to work on a guidance that will be dedicated on lipid-based nanoparticles for RNA delivery. So I want to point out the fact that, of course, regulation it was lower. It took a long time to get in, for example, for liposomes, but now they are speeding up. They are aware of what is the need, and they are also seeking for advice from different experts in the field, and they are moving. So I really hope, and I'm confident that with the new innovative formulations for delivery of biologics, the process of getting new guidance will be faster. And we know that they are working on that, and so they are also adapting, and they are very well aware of the needs.
Thank you for your comment, Fanny.
Yes,
Very
no,
welcome.
that's very useful. And I think, like as I've mentioned, I think some of, the way the regulators learn, very often they've got more information clearly than, most of us as individuals have or individual groups have. So I think it is, it just emphasizes the need to go and talk to them a lot earlier, because that's what's there. There's another question here. Perhaps we can address this. That's coming from Nicholas Carvu. It's how important is imaging at the translational level? Is it more or less important than that of proof of concept? So Fabrice, any thoughts on that one?
Imaging, I have the same opinion than Patrice on that way. It's also the link with your previous question. So the evolution of recommendation by regulatory agency, but also the evolution of methodology. So we need also characterization of the nano product. That sometimes we do not have the biological assay to do that. Follow some nanoparticles, especially the natural nanoparticle, or what we call the synthetic one, by using bioingredients. You put this in the blood, and sometimes you cannot follow the particles. So imaging is good. Sometimes it's very useful, especially for inorganic particle, because you can follow the iron oxide particle in your body, even in humans. But if you work on liposomes, if you work on polymer particles or other kind of lipid nanoparticles, it's very hard to follow them in biodistribution, imaging at the human level, or even at the preclinical level. You follow the label, you follow the dye, but you don't follow really the particle. So you have to invent new methodology to do that. It is not so easy. That's my comment.
Thanks. Thank you. Patrice, any follow-up to that?
No special comment. I mean.
Okay. Thank you.
Can I ask a question, Alan?
Yes, of course you can.
I catch Venner here. Would it not be possible to simply tag the nanoparticles by itself and have a biodistribution analysis being done with the animals, sacrifice them and identify where they are in the organs?
Okay, if you tag it, it's no more the same particle. So that means the tag itself may change the behavior. It's always the nightmare for that. Unless your nanoparticle contain a contrast agent and is part of the nanoparticle. But every single change in your nanoparticle, you build a new object.
Yeah, I understand that. I understand, but at least it could give you a hint in terms of biodistribution.
Sure. But again, in terms of regulation, I'm not sure that it's completely relevant. In terms of science, we can always discuss and say, I have a good reason to think it is the same with the untagged nanoparticles.
Yeah, okay. But I've been, when I have started my work in pharmaceuticals, I was working on carbon 11 labeled nimodipine. Those type of things are possible, aren't they?
Sure.
Or carbon 13 labeled, whatever.
Yeah, sure. Yeah. Radioactive or isotope labeling is probably the milder, I would say, marker that we can use in terms of chemistry.
Yeah, but
exactly.
You have to combine the labels because you have several components in your particles, which are stable in the form of the particle. But you have to be sure that all the labels are in the same place to say that you follow your particles themselves and not the degradative products.
Yeah, that's a good point. That's
sure,
very
I
understand.
I understand, but then you have to define or invent several labels across the whole micelle or whatever you have. Okay.
Yeah. Daniel, any comments on that one too?
Just to say that it's challenging. With inorganic, it's quite easy. Okay, we have experience with that. So you extract easily the component, and then you with analyzers you detect the inorganic component, and then you have where the nano particle is where, and also the degradation of inorganic nanoparticles also is different, and is with the shell life is also longer. So stability and cell life also is something to have into account, and it's a higher level of complexity.
To have a challenge is nice, isn't it?
Alan, may I add something?
Yes, of course you can, Ruth.
Yeah, I think as long as the nanoparticles are not body-own compounds, today you can really do very nice biodistribution studies with using MS, which is a label-free technology. So we do quite a lot of biodistribution studies now in several projects where you can follow the drug and you can follow components of your nanoparticle. Even degradation products, as long as these are not endogenous compounds.
Okay, thank you. Thanks for that. Patrice, can I bring you in now to talk about immunological considerations? I mean, because we're dealing with more biologicals and the way they're working, and we need to think about immunological responses now very early on in a drug's development life. What are the considerations you take into account and have to think about when, from an immunological point of view, in developing a nanoparticle and a nanomed?
This is an issue which is really under development, and it's really raising concern. It comes from the simple fact that immune system is the guardian of the body. And it comes back also to the point I forgot to tell you before. I come back to my mind is when you deal with drug, you deal essentially with soluble molecules. When you deal with nanotechnology, nanomaterials, you deal with particles, and the change in the scale involve a different behavior of the immune cells. For instance, the first target of nanomaterials or nanoparticles will be the phagocytic cells. That's the macrophage, the neutrophil, and the dendritic cells, which are specialized to engulf any foreign materials coming from outside. And we know now for plenty of different works of different with different nanoparticles that they accumulate in those cells. And one of the first question is the acute reaction, because those cells are supposed to detect any intruder and to deliver danger signal to the immune system and raise inflammation and an immune response. So that's the first point. And the second point is a long-term response, since those cells accumulate these materials, and these cells, like macrophage, can last several decades within tissue. So that means that they can survive a long period of time with the material inside. So if there is any effect, it could be long-term effect and could lead to chronic disease like chronic inflammation, which could be not by themselves create a disease, but can be a comorbidity factor, favorizing emergence of autoimmunity or chronic inflammation and so on. And of course, the issue of long-term effect of nanoparticles is one of the most difficult to tackle, because it's difficult to settle experimental work on exposure for 10 years, for instance, to some nanoparticles. Is not feasible in our short life of scientists. But it's a really concern, because if you develop a drug and you spend a lot of time and you invest a lot of money, and you discover after 10 years that your drug is provoking a chronic inflammatory disease, you may disqualify your drug, and it could be very expensive for the company.
Yeah, Alan, I think that Chantal, you have a comment to do?
Yeah, that's fine.
I see that you raise your hand, so I mean that you want maybe to tell something, Chantal?
No, Chantal, can you speak yet?
Can you hear me?
Yes, can, Chantal. Yes.
No, I didn't want to speak, but it was interesting, this point, and yes, I agree with Patrice on the particle aspect of this material, which is very different from soluble molecules of the immune system.
Okay, thank you. I mean, one follow-up question I was going to ask Patrice. I mean, given that there are now just over 50 nanomedicines that's been approved, is there much evidence that this is causing immunological effects in the patients using these treatments? You know, because some of them have been out now for a few years. And are we seeing anything, are we aware of any particular safety issues that are there with these patients that they've experienced due to the immunological effects? So, I mean, you know, one thing that springs to mind, you know, are there sort of neutralizing antibodies that knock them out, for instance, and things like that?
Okay, so for instance, the story about there was one of the concerns at the very beginning. We were concerned by the possibility to have antibody against the product, like we have some on soluble molecules, like for instance, biological protein, which are recombinant protein, which are used for medicines. Actually, there is very few examples of immune response, specific immune response, antibody response to nanomaterials. It's apparent not to be the main concern in that field. So as far as I know, but I will be interested if anybody has a specific comment on that. So that's
yes. Yeah, so clearly isn't a literature on it yet from what you've said, particular literature. So it's probably simple single cases, is it, or small reports about it with what's going on. So, okay.
Unless we refer to the complement activation, that's something.
That's something else. I mean,
that is
a
different kind of immune reaction, of course.
You're absolutely right, Raymond. It's very important. It's even mandatory to test for several assays like complement activation, which can drive a strong inflammation in the acute phase. Essentially, in the acute phase, it's one of the very first assays that Daniel will do when he will receive some nanoparticles, and hemolytic assay also there is a list of some assay, but that's essentially on the acute phase. So I think for the acute phase now we have tools. It's okay right now. What is much more difficult and much more challenging is to guess for long-term effect, long-term exposure, and the effect at long term.
Okay, well, that leads us quite nicely into Daniel and my question to him. You know, in terms of the preclinical work and assessments, what do you need to consider in relation to nanomedicines when you get involved? You knew this was coming.
Yeah, you let me 2 hours, no? Well, what's so big question? It's first, when we receive nanomaterial, what we ask is to have in mind that any modification should be done at the beginning, because if you change the charge, if you change the components, you have a new element. So it's very, very difficult for us to perform the follow-up of the improvements of the nanomaterials. So the first thing we need to consider is how well established is the nano component. And once they are sure that this will be the final component, they need to provide us a way to characterize this component and check how this component is changing, because the stability is one of the first concerns of nanotechnology. So how long the nano component is stable and on time, and this is maybe the first problem we encounter in our lab is to receive nanomaterial, and they told us, okay, it's stable 24 hours, so it's not feasible. This is the first thing that we are focused in, is in how we can measure how we can characterize something that is not stable and during time.
From your experience, do you identify any particular signals from the toxicology work that then has implications for following it up when we get into humans? I mean, you know, obviously with new chemical entities and chemicals and the biologicals anyway, you know, we will pick up signals. But is there anything specific to nanoparticles that causes signal to be followed up in humans and the use of the nanoparticles themselves? From what you know?
Well, for us, the first difficult thing is the sterility, how the material is sterilized, and there is also the endotoxin content of the nanomaterial. So sometimes we have some side effects, we have some immunological response. It's not due to the nano component, it's due to the endotoxin content and also the burden that can be in the nanomaterial. So this is very important and challenging, how we get the product sterile, how we can sterilize this material, and how we can be sure that it's endotoxin free. And this is very, very challenging to try to detect this endotoxin, to quantify this endotoxin, and then to find the correct I would say levels that you can inject to animals.
Thank you. Thanks. Now we've got about just under 10 minutes left. If anybody's got any questions from the audience, then please do type them up or put your hand up, and we can get you answering and things.
I have to comment for you, Alan, and
yes, of course.
The attendees. So, on the matter of distribution of study of the impact of nanoparticle on immunology, I think it's very important to have this in mind at the early stage of development, because you better know your products, you better know also its potency, its potential as well. So by studying the biodistribution, you know where you can deliver a drug. So that also gives you some ideas to develop new therapeutics. But it's also the case for immunology, because you can interact with the immune system, okay, because it's a very sensitive system, and Patrice is there to comment that. But you can also play with that to develop your therapeutics. That can be a vaccine, that can be commented by Patrice, but also in the case of if you have an impact on the inflammation, you reduce the inflammation, you can also consider maybe anti-inflammatory strategy. So I think it's very important to have this in mind and all the studies on that to design your product.
Yeah.
There is one point that should be taken into consideration and to be retained. Any nanoparticle injected in the body will accumulate in macrophage and phagocytic cells. Wherever you want to target them, in the brain, in the kidney, in endothelial cells, or whatever, once you inject them, you are sure that most of the material which will be delivered in those immune cells. So that's mean it's you cannot avoid analyzing the fate of these cells once you inject a particle.
And I have a question for Daniel and Patrice concerning the study on immunity and immune system. What is, in your opinion, the long-lasting effect we can have on the immune system by working on nanoparticles? And more especially if we consider to repeat the doses and the injection of the nanomaterials.
Yeah, well, I mean, as I said, it's essentially on the phagocytic cells that we may, for long term, because they retain for long time the materials. But you know that you can change the metabolism of those cells. This is there are several studies which show that after you phagocyte the cells are phagocyted, nanomaterial, they may change their metabolism, and since the metabolism is controlling the pro-inflammatory reaction, you may then switch your macrophage or your cells to a pro-inflammatory signal. So this is essentially. Then the issue depends where is your immune cells and where is the site of injection disponibility. But you know, chronic inflammation are involved in so many diseases that you open the door to you open a Pandora box. You know, this can be anywhere.
Yeah, it's the fact. So the chronic aspect is the most relevant. So even if the pro-inflammatory effect is small, the thing if the treatment is chronic, then you have this inflammation once again, and the effect can be accumulative, and then induce other sensitivities, and then you will change your basal response, response, for example. So at the end, when there is no problem, everything is okay. But since you have illness, so this background basal inflammatory can conduct to a grave problem.
Let me ask a last comment. It's not because I don't want that the people get out from the workshop, from the meeting with a pessimist view. It's not because your nanoparticle will generate a somewhat pro-inflammatory effect that your nanoparticle will be disqualified and not useful. You have to consider that in the body you have also reaction against inflammation. So that it is just a balance. If you want to have an example, just follow what's going on with the COVID infection, where sometimes you have an asymptomatic phase, and sometimes you have a huge inflammation which causes dramatic dramatic evolution and death. So we don't know that the body and the immune system has its own capacity to counterbalance any inflammatory signal. But then what is important is that actually our role in doing this is to warm the developer, to say, hey, pay attention to that point because your material may promote inflammation. Then consider it properly into this development to master and to follow that. That means in the clinical trial, maybe introduce some assay to follow if you create really inflammation in vivo. Into the patient. So don't be pessimist if you have a sign of inflammation in vitro, but be informed, be aware, and adapt your strategy of development in consequence.
No, thanks for those comments. Fabrice, you got a comment?
No, I see that there is a question on the chat. So maybe you have seen this, Alan? Or?
Yes, I'll read it out. So the question from Marco Monopoly. Testing the efficacy of a new nanocompound is quite challenging. We often refer to active targeting or passive targeting by the EPR effect, and not all tumor types have the same architecture and response. So I think what we're talking about here is, in cancer. Do you think that our community has enough interactions with the real world, i.e. a pathologist, etc.? So I think it's a good point, is actually involving, and going out, and because clearly, from experience, tumors do mutate, they do change, so how are we adapting to that with how we're using nanoparticles and nanotherapy around that? Anybody got any comments on that one? We may want to open it up to other people.
I think it's a matter of preclinical experimental cancer in mice, for example, but it's not only related to the EPR effect. It's also related to the interaction with the immune system. The immune system of the mice is not the same that from the humans. So I think it's related to the preclinical models. It's a huge topic.
Yes. We probably could have a whole workshop on that ourselves, couldn't we, actually? But perhaps we'll come back to that and get other people's comments. So we've got a couple of minutes left. Just ask, sorry, there's another question come in here. So if we suspect that our nanodrug may be causing inflammation, how do we best evaluate it? Probably for both Patrice and Danielle.
Well, it depends then after the context of the treatment and where you will deliver your materials, in what organ, and you may then after design with the pathologist, as we said before, with the clinician and with the regulatory affair, some essay to follow the status of the inflammatory response. So it can be done systematically on the analysis of immune parameter and inflammatory parameter, either locally or systemically.
Okay, thanks. Well, I need to draw this to a close because time's getting on. Danielle, any final words from you at all?
Maybe the first advice is have everything in mind since the beginning. And guidance are welcome. Everything is welcome, but the feedback, the multidisciplinarity approach, it's what at the end will give the success of the investigation. Surgeon feedback, everything is very important, but things should be taken into account since the beginning and not at the end, because it's so give more importance to the preclinical and the research before go ahead and make the next steps.
Okay, thank you. Patrice?
Nothing to add, because it's exactly what we should promote and support.
Yes. Fabrice?
No, I do agree that we have to work together. And also to include at the early stage regulatory expert, because we have new therapeutics enter into the market. So this opens the way of a lot of methodology and regulation to take into account. So we have to follow this from the beginning.
Yeah. Okay, thank you. And final words from me. I know we haven't actually spent any time at all talking about clinical studies and the design of those studies. But I mean, certainly with what we've done with the New Deal, we started designing the first in-human study, 2 years ago, even though, we're a long way from that. But taking that back with the target product profile and what we want to achieve with the product, we've worked backwards from that about how we then design not only the first study, but what the implications are for the toxicology, immunogenicity testing, and the manufacturing. So it's key that we do that. In fact, designing the clinical studies is like for any other indication, any other therapy. There are not particularly special things that we do differently. We'll still have to decide and choose what dose we go forward with, which, certainly in the New Deal program, we haven't done that yet. But that's really where we are with that. So I'd just like to thank my colleagues, Patrice, Daniel, Fabrice, and everybody else who contributed and spoke up. And